Hello guys and welcome back to Excelsior Academy. So in the yesterday's lecture, we just started with the DNA replication and we talked about the basics or the chemistry behind the DNA synthesis, right? So in today's lecture, we'll talk about the DNA polymerase. So we know what does DNA polymerase does? It is mainly responsible for your DNA replication. Okay. So first we'll talk about DNA polymerase only, like how it is exactly, catalyzing the reaction. What reaction? The reaction of DNA synthesis. Later on, we'll talk about the types of DNA pol. Okay, so first we'll talk about only DNA polymerase, like how it is catalyzing the reaction, how it is giving the proofreading mechanism, what all additional characteristics are present in the DNA polymerase. Then we'll talk about types of DNA pol in prokaryotes and eukaryotes. Then we'll talk about the different enzymes which are present in the DNA replication, and then initiation, elongation, and termination of DNA replications. Okay, so with that, the DNA replication topic will be clear. Okay, so now let's talk about the DNA polymerase. So it uses single active site. Okay, so here only one active site is there, and that is what we are going to discuss in today's lecture. So it is catalyzing this by exploiting the nearly identical geometry of the AT-GC pairs. Okay, this is AT and GC pairs. So what does DNA polymerase does? It is going to monitor the ability of the incoming nucleotide. So incoming nucleotide is your DNTP, okay, to form ATGC pair by rather than detecting the exact nucleotide. So how it is happening? So in the, okay guys, first tell me where does the DNA replication takes place? In the cell? Yes, Shruti?
Nucleus, ma'am.
Yes, very good. Yes, it is nucleus, right? So in the nucleus, you have got the DNTPs pool. So what do you mean by pool? Means so many different types of DNTPs are present for G, for T, for C, for A, right? Now what happens is that this is going to be a chance event. Like active site में आपका पहले G आना चाहिए, T आना चाहिए, C आना चाहिए, A आना चाहिए, ये आपका chance event है, ठीक है? Whenever it is the exact, like A के opposite में T आना चाहिए, जब भी T आएगा, then only the bond is formed. But if suppose G is coming, it won't be able to sit in the active site pocket. So it will go back. Then A will come. A is again not able to sit against the A. It will also go. Similarly with C, only T is able to sit against A. Then only the DNA polymerase is going to make the bond. इतना समझ में आया?
Yes, ma'am.
Because rather than detecting the exact nucleotide means it's not possible for DNA polymerase to go and select from the pool, like against A, T I should pick, or against G, C I should pick. DNA polymerase can't do that. Okay, so you have got the DNA, the primer is there. Okay, DNA polymerase is there. Now what happens in the pool? Randomly DNTPs are coming. So whenever correct DNTP is coming and sitting in the active site of the DNA polymerase, then only the bond is formed. Otherwise, it is being removed. Okay. Now this is happening like in fraction of seconds, like 10 raised to minus 8 seconds. So this is very fast event. Okay. That's how your DNA synthesis is taking place. Otherwise, if this process is taking time, what will happen then? We will never age, because the DNA synthesis will be very slow. We are not going to form the different cells. So it will take, million years to become a complete adult. But we know that is not happening, right? We do die after 70, 80, or maybe 90 because of the aging and all, if at all accidents are not happening. Okay. Okay. So only when a correct base pair is formed at the 3-OH of the primer, the alpha phosphate of the incoming nucleoside triphosphate in the optimum position for catalysis to occur. Now this will be very clear once we see the figure. Okay, look at the figure now. Now, see the correct base pairing. Are you able to see? See, this is how the template is going to be. Okay. This is your template. This is your primer. Primer ka 3-OH. So to this only your new NTP, new DNTP will come. Okay. This is like this. Just imagine like this. This is your template. This is your primer. Okay. 5-, 3-, 3-, 5-. Okay. So this is your OH over here. So now here new NTP is coming. DNTP is coming. So on the template we have got GC. So here GC, here A. So T should come, right? So now D, T, P is coming. Now this much is clear, guys? Yes? Okay, so now see. So in yesterday's lecture, we talked about how 3-OH, the oxygen is attacking at the alpha phosphate, right? So we discussed about the SN2 mechanism. Okay, now you can see over here, same thing. The 3-OH is attacking at the alpha phosphate. Okay, now what is happening? Then it is possible for the DTTP to sit in the pocket. Can you see it? It is able to form. So first what happens is, first they make the base pairing. Okay, so A के against में T आपका base pairing हो गया, correct. Now, because of this correct base pairing, OH of this ribose of the incoming DNTP is able to sit in the pocket, correct. Then only the 3-OH is able to attack at the alpha phosphate. Then your bond is formed. Okay, so what are the 3 steps that should be taken care of? First is the base pairing. Okay, if suppose यहाँ पे G होता तो यहाँ पे C के साथ पहले वो base pair बनाता, then only ribose which is present of the C should OH. ये कौन सा OH आ रहा है यहाँ पे? Can you tell me? This is 2-deoxy. Okay, तो यहाँ पे सिर्फ H है. This is a 3-OH. Now, so what is happening? This 3-OH should sit in the in this pocket here. So this is second condition. Then only the 3-OH of your previous nucleotide from the primer is going to attack at the alpha phosphate. Okay. So yes, guys, tell me, is it clear to you everything? Yes, Akshaya, clear हुआ थोड़ा?
Yes, ma'am.
Huh? Now who can tell me? Shruti, can you repeat what I have just said? What are those 3 conditions or steps for a correct nucleotide matching?
Yeah. So first, the right nucleotide has to be at the at that particular position, and then the OH has to fit into the right pocket, and only then the next nucleotide can join in because of the position of the OH. Like then all 3 of them are in the right position, and then the bond is formed.
Okay, it's half partially correct. Shruti, how we have to say is that first correct base pairing has to form. Second, second, the 3-OH has to sit into the binding pocket. Okay. Third is primer ka 3-OH is going to have going to attack on the alpha phosphate for the phosphodiester bond formation. ठीक है, बस इतना ही बोलना है. Okay. Okay. Now coming to the correct base pair, like how DNA polymerase is able to understand, like not to join or not to form a bond with the wrong nucleotide. Okay, so for this, again, see, A is not able to form the base pair with the another A. So because of this, eventually the OH of the incoming nucleotide is not able to sit into the pocket, the binding pocket, right? Hence, this is not aligned properly. The alpha phosphate is not near to the 3-OH of the primer template, right? So this is also not formed. Hence, the bond is not formed. So this is how the incorrect base pairing is not possible in the DNA polymerase. Is this much clear to everyone? Yeah. Okay.
Yes, ma'am.
Now, so incorrect base pairing leads to dramatically lower rates of nucleotide addition as a result of the catalytically unfavorable alignment of the substrate. Okay, so this is an example of kinetic proofreading. Now, this is also important to understand what do you mean by kinetic proofreading. This is for the MSc students. So, in what does kinetic proofreading means? It means in which the enzyme favors catalysis using one of the several possible substrates by dramatically increasing the rate of bond formation only when the correct substrate is present. So DNA polymerase have got high kinetic proofreading because incoming nucleotides will be coming. Okay, so very fast, just based on the correct base pairing only the bond is formed. Okay, so the among all the 4 different DNTPs, DNA Pol is able to pick the correct nucleotide based on the correct base pairing. Okay, so this is kinetic proofreading, and later we'll see that how fast DNA polymerase can carry out the replication. Now, how does DNA polymerase can distinguish between RNTPs and DNTPs? So, what is the difference now? Here, in the structure, we have got, see, this is just sugar, ribose sugar, right? Excuse me. Excuse me. Okay, so here we have got the bases attached. You can have A, G, T, or C. Then this is your 2-OH, 3-OH. So this is in the case of your RNTPs, okay, ribose, which is used for your RNA. Now, in the case of DNA, you don't have 2-OH. We have 2-H only, and that's why it is deoxyribose, right? Now, how based on these ribose and deoxyribose, DNA polymerase is able to distinguish between the RNTPs and the DNTPs. So this discrimination is mediated by the steric exclusion of RNTPs from the DNA polymerase active site. Steric exclusion क्या होता है? Because of the heavy groups, it is able to expel them out. ठीक है, उसको धक्का मार के निकाल देता है just because of the bulky groups present. So in the DNA polymerase, the nucleotide binding pocket cannot accommodate 2-OH on the incoming nucleotide. So अभी हमने previously क्या देखा था? That it has got the pocket available for the 3-OH, okay, but 2-H के लिए. So it allows 2-H but and 3-OH, okay. But if you have got 2-OH and 3-OH, which is present in the case of the ribose RNTPs, then it is not possible. Because see, ये क्यों जरूरी है? क्योंकि nucleus में along with the DNTPs, you also have RNTPs. We have got so many proteins, we have got so many sugars present in the nucleus. So randomly कुछ भी आ सकता है. But since they have the difference only at the 2-H and OH, it becomes crucial for the DNA polymerase to identify the correct one. Okay. So this space is occupied by the 2 amino acids that make van der Waals interactions with the sugar ring. Now, what amino acids? Amino acids of the DNA polymerase. Okay, so instead of accommodating 2-OH, they have got the amino acids taking place over there, and they will interact with the ribose. Okay, the sugar over here is the ribose with the help of the van der Waals interactions. Guys, this is important. Okay, this slide is important to understand. Okay, so let's look it over here. Now, This is again the same. You have got the template, and this is your primer, 3-OH, again attacking. Okay. Now, in the previous figure, we saw that it is making a correct base pair. Okay. So it is going to make a correct base pair first. This was the first condition. This correct base pair is going to allow your 3-OH to sit into this pocket, right? And then only the primer, then only the 3-OH is going to attack at the alpha phosphate, because the alignment is going to be the proper. Okay. Along with this, we have got additional, the fourth condition is that we have discriminator amino acids. This are going to distinguish between RNTPs and DNTPs. Okay. See, so what they're doing, in the case of correct, you don't have OH. Here, you don't have OH. What is present over there? The 2-H. Okay. So they are not taking the space. There is no steric clash. There is no, I say, the OH of the ribose is not, it's not fighting for the space with the amino acids which are present over here. Okay. Excuse me, right? Now, coming to the incorrect one. So, A and U. So, what is U over here? The RNTP. It has got 2-OH. Now, this 2-OH is fighting for the space which is present over here. But already their amino acids are present. So, what will happen? It will not allow the accommodation of the 2-OH at the discriminator site, and because of which there will be wrong alignment of the phosphate group, and hence the OH is not able to attack on the phosphate. Because see why it is important, the discriminator amino acids, because here the base pairing is happening correct. We know that A also forms 2 base pair with the uracil, right? So the OH should sit into the binding pocket, but in the DNA polymerase, you can't have RNTPs. In order to avoid, we have got the additional. Okay, so that is your discriminator amino acids. Yes. Chandni, is it clear to you?
Yes, ma'am.
Yeah, everyone. What about you, Tanvi?
Yes, ma'am.
Okay, nice, good. And all these figures, they have been taken from the Watson reference book. Okay, so if at all any doubts are there, you can go back and refer the Watson. Okay, now coming to the DNA polymerase, it resembles like a hand which grips the primer and primer template junction. Okay. Okay, so let's now talk about how the DNA polymerase looks like. So this is how it is going to be. So the gray colored one is your template and the green one is your primer. And this is how the DNA polymerase looks like. Okay. Like actually, frankly speaking, when I saw the 3D structure of the protein, the 3D structure of the DNA polymerase, to me, it does not look like hand. I don't have that good imagination. But for the crystallographers, the protein crystallographers are there. When they saw the structure, for them it was like a hand. Now, why they're saying hand-like structure is that, see, the DNA polymerase has to move on the primer template, right? It has to move as it is going, keep on adding the DNTPs, right? So this is happening in the palm region. In the palm region, the DNA synthesis is happening. The fingers and the thumb, okay, they are responsible to make sure the correct nucleotide is coming, then there is a proofreading. See, sometimes what happens is that there might be wrong base pairing and the nucleotide has been synthesized. So what will happen if suppose there is a wrong nucleotide has been added? You have got the template like this, okay, and there will be bulge out like this. Since there won't be any base pairing, so there will be bulge out. So this bulge out will be again scanned by the DNA polymerase, right? This is like agar aap rope mein aise haath le ja rahe ho, so suppose there is something in between, you will understand. In the same way, when DNA polymerase is scanning the DNA strand, if there is something bulge out, it understands that something went wrong. That is, wrong base pairing has happened. Okay, so wrong base pairing has happened. So what should be done then? Then it should go there, make a cut, remove this DNTP, and then add new DNTP, and then again seal it. Okay, so this we'll study now. Okay, so we got all this understanding with the help of the X-ray crystallography. Okay, so this is also important, guys. Now, whoever is working in the field of protein, cancer, or diabetes, like whoever is working on proteins nowadays, all over the world, to understand their function. Okay, now many people are not doing any biochemical or biophysical activities. What they do is they go for the X-ray crystallography. There is something called SAR. Can anyone tell me what is SAR with respect to protein or enzymes? It is your structure, activity, relationship. Now, why I was asking this is because based on this only the drug or the vaccine against COVID was made. Now, I'll just give you a small hint like how it is being done and all. See, once you have the structure, okay, now once you have the structure, the active site, the catalytic site is known to the scientist, okay. Then once the active site is known, then you can design the drug to inhibit the active site of the protein, right? So with the help of the structure nowadays, if suppose you have got these something structures like this at the active site, so you need to make a drug which can sit over here and block the enzyme. So this relationship of the structure activity is helping us to design the drugs. Okay. Now, in the case of COVID-19, in India, many people have used Covaxin and many have taken COVID shield. Sorry, shield, yes. Now, can anyone tell me the difference between the Covaxin and the COVID shield? ये तो बहुत latest है, so you guys should know. Yes, anyone? Chandni, what about you?
No idea, ma'am.
What about others? I think you guys have taken the shot, right?
Yes, ma'am. Yes, ma'am.
Now for
Yes,
the
Covaxin
yes.
is live attenuated, ma'am.
Covaxin is live attenuated. Sure.
No, no, it's whole killed.
It is whole killed. Yes, very good. It is whole killed. And Covishield?
They use the spike proteins on the adenovirus.
Yes, so they have taken the spike proteins and they use the technology of CRISPR-Cas. So this is happening at the mRNA level. Okay, but this is like normal, जो हम हमेशा से use करते आ रहे, like at the time of Edward Jenner and Louis Pasteur. So this is like thoda sa hi-fi fun. We'll discuss about like CRISPR-Cas and mRNA level, how they have done and all. It's a very long procedure. Okay, so that's why many people were hoping that, thinking that COVID shield is more effective as compared to Covaxin. But later on, Covaxin was also found to be effective. Okay, because see, whether it is spike proteins or the DNA or RNA of the COVID-19 virus, entire whole killed virus was injected into the human body, right? So the antibodies was produced towards the total virus. So we are going to be safe. Okay. The only thing is, like, you must have heard that even after getting the vaccination, people are getting affected by the corona. They are getting positive. So, but able to fight back the problem. Okay. So, whole kill and mRNA level CRISPR-Cas. So, we'll discuss this. Good. This much knowledge should be there because see, now corona has impacted everyone's life a lot, right? So, in future, now this is going to be in your textbooks. This is going to be for your exams also. So, definitely it is there for your interviews also. So, you should know at this stage about the COVID-19, why it is COVID, why it is 19, whether the genetic material is RNA or DNA, whether it is single-stranded or double-stranded, okay? The life cycle of COVID-19 and the drugs in the market used, drugs or the vaccine which is used in the market, okay? So if you remember when this pandemic started, when vaccine was not there, what kinds of drugs were used? So they started with something called hydroxyquinone, if you have heard in the TV and all. So that drug is mainly used as
Malaria.
anti-malaria. Yes, very good. Okay, but people have shown that it has got the resistance and all, and I don't know. And India was exporting this medicine to different countries, right? Because we have got the source. So we were exporting all those. Anyways, so now vaccine is there. Now, before moving on, like I just want to check like how much thinking you have. So we have taken the drug first was given to the old people, right? Then to the 30 to 40 wala range wala people, like the working people. Now they have come till 15 to 18, right? Why can't we give this vaccine to everyone? Why can't we give it to 0 to 2 years of age children? They're again still in the threat region, right? Why can't we give vaccine directly? Because already from their birth till 2 years of age, they are taking so many vaccines. Why can't we give it them now? Why are we waiting? Because the work is still going on to make the vaccine for the different age group. Why is it so? Why they started with old people and then this people, then this people? Can anyone tell me?
I think it was related to the amount of immunity that the general trend showed, as well as the level of protection these people required. Like old people are more likely to have more comorbidities, and therefore they needed to be protected the most. And 0 to 2 years, the immune system isn't completely developed, so they had different requirements.
Yes. So we don't know how this is correct. Like old people, they have less immunity and all, and the risk of them getting the corona was high, and plus they don't have that much immunity, the energy to fight back. That's why they were going till ventilator, ICU, and it was difficult for doctors to save them. We have seen many cases where old age people were dying, right, when the pandemic started. Many people were able to recover at this age, and but many people they were not. So it depends again on the immunity level, like your lifestyle. Now they have given for the 15 to 18 also. Now 0 to 2, why? Because we don't know how this vaccine is going to react in the body of the babies or the kids, right? We don't know, and it is not tested. Like the clinical trials have not been done, right? And plus, vaccine means giving them the immunity, the chances. So over period of time, we know how polio vaccine is working. We know how this MMR, BCG, everything is working. It's already like for the many years we have been using. Okay, so we know the mechanism. We know how it is going to react in the kids' body. We don't know anything about corona or COVID-19 in the case of 0 to 2. That's why it's not been given. And people said that be careful. And that's all. Good attempt. Okay, let's come back to the topic. Okay, guys, just join me back because the meeting time is getting over. We'll continue from here itself. Okay, just join me back. I'm stopping now. So we were talking about how the X-ray crystallography is helping in understanding the DNA polymerase activity. Okay, so these structures reveal that the DNA substrate sits in the large cleft that resembles a right hand. Okay, now based on the hand analogy, see guys, this is how you can remember also. Like looking at your own hand, you can remember what does fingers, thumb, or palm is doing as the DNA polymerase. So the three domains of the polymerase are thumb, the fingers, and the palm. Okay, so the palm domain is composed of, this is beta sheets, which contains the primary elements of the catalytic site. So of course, looking at the figure, you can say that palm, the catalytic site is over here. So here is the basically your main DNA synthesis happening. And the recently synthesized DNA is associated with the palm, and the site of DNA catalysis is located at the crevice between the fingers and the thumb, like over here, this region. This region. So the single-stranded region of the template strand is bent sharply and does not pass between the thumb and the fingers. Okay. Now, can you see in the structure how nicely over here the the helical structure is there of the DNA, but over here, can you see there is a bend? Yeah, can you see the difference? Can you see the difference over here? Like the entire helical structure, and then
Yes.
over here it is a bend is present,
Yes,
right?
ma'am.
Yeah, so this bend is actually helping. This bend is actually helping for the corrective base pairing, which is happening in the palm region. Okay, we'll study more in detail. Okay, now the metal ions. This is also very important because see, this will tell you why in PCR we need, if any of you have done PCR once, you know that we need Taq polymerase, of course. We need DNA template. We need primers, both reverse and forward. We also need buffer solutions as well as MgCl2. Okay. So now we'll know why we need MgCl2. So the palm region of DNA polymerase, it binds to divalent metal ions like Mg2+ or zinc that alter the chemical environment around the correctly base pair DNTP and the 3-OH of the primer. So now we know that the DNA polymerase requires Mg2+ in order to catalyze the activity of the DNA polymerase. Okay? So that it can form a phosphodiester bond. So one metal ion reduces the affinity of the 3-OH for its hydrogen. Now, why this is required? To reduce the affinity of 3-OH for its hydrogen. Then only this oxygen will be free. See, OH stable है. अगर मैं H हटा दूंगी, what will happen? This become H plus and O has the, oxygen has the negative charge, right? So this generates 3-O minus, which is now primed for the nucleophilic attack of the alpha phosphate of the incoming DNTPs. Remember? So we were talking about 3-OH attacking at the alpha phosphate, right? तो ये कौन initiate करेगा? आपका Mg2+. How? O minus OH में what will happen is, once the electronegative atom is losing the affinity for the H, what will happen? They will be removed like this. ये H plus release हो जाएगा into the pool and O minus is still attached. Now this is O minus. It needs more to become stable. So what will happen? It is going to attack at the alpha phosphate. ठीक है? Now the second metal ion. So there are two which is required. So एक का काम है to reduce the affinity of the 3-OH. And the second one, to coordinate the negative charge of the beta phosphate and the gamma phosphate of the DNTP to stabilize the pyrophosphate, which is produced by joining the primer and the incoming nucleoside. See, this is why I'm saying all the lectures are important because they all are connected. So in the previous, we talked about 3-OH attacking at the alpha phosphate. Okay. And the bond is being broken. So aapka beta and gamma needs to be stable. They are also highly negative. So that needs to be stabilize. So this stabilization is done by the second metal ion, or that is Mg2 plus. Is the theory clear to you? Then it will be more easy for you to understand the diagram. Is this much is clear? I'll just summarize the slide. So you need two divalent metal ions, okay? That is Mg2 plus or zinc, which is helping in the phosphodiester bond formation by the DNA polymerase. Okay, so first metal ion is reducing the affinity of the 3-OH. Then only the 3-O minus is able to attack on the alpha phosphate. Second is stabilizing the beta gamma phosphate, which is the pyrophosphate, जो हम PPI लिखते हैं. Okay, so this is being stabilized. Later on, your pyrophosphatase will come and convert your PPI into 2PI. Guys, this much is clear?
Yes, ma'am.
Yes, ma'am.
Yes. Now look at the figure. So here also the same thing is being we have tried to explain. Again, this is your template primer. Okay. Now look at over here. Can you see the metal ions? Two metal ions. They have been mentioned as A and B. Can you see it over here?
Yes, sir.
Yeah. Okay. Now also try to understand here. They are making bond over here between C and G. Okay. So correct base pairing has been done, C and G. The OH is also sitting at the binding pocket, and the 3-OH is now aligned to attack at the alpha phosphate. That is your first one, alpha, beta, and this is the gamma phosphate. Now you need to stabilize the correct incoming DNTP, and that is done by the metal ions. So this is now go towards the left hand side of the DNA polymerase. अभी तक हमने सिर्फ इतना देखा था, right? So now we are looking at this portion also. So two metal ions are required, okay? And look at here. So first metal ion, which is represented as A, इसका क्या काम है? So it is going to interact with the 3-OH. So it is resulting in the reduced association between the O and H. See, किसी negative के पास already एक positive था, अभी दूसरा highly positive आएगा, so what will happen? This is only plus, this is double plus. So what will happen? This plus is losing its affinity for the minus. So ये H plus जो है आपका चला जाएगा, ठीक है? Now this is being stabilized over here. Then because of this association, this O minus is going to attack at the alpha. Okay, it is making as a nucleophilic 3-O minus. Now second, metal ion B, what it is doing? It is going to stabilize your beta gamma phosphate. Can you see how it is beautifully interacting with the oxygen over here, which is present, right? Clear हो रहा है आपको? Now, one more thing you can see in the figure is the presence of the aspartic acid. Now, why this aspartic acid is required? 2 plus है, एक oxygen की तरफ जा रहा है, 1 is also there. So even that needs to be stabilized. Then the aspartic acid, it is a amino acid, right? So it has got COOH. So that is going to interact with the another plus. Okay. So overall, these are helping in stabilization of each other. Okay. Any doubts over here? Should I explain again?
Ma'am, can you repeat about that aspartic acid?
हाँ. Okay. So इतना clear है that the metal ion is O minus, यहाँ पे plus है, so they are interacting. Okay. So only the plus, there are two plus, right? Plus and plus. Mg2 plus is there, right? Just think like a mathematical. So एक plus है जो आपका O minus की तरफ contribute कर रहा है. There is another plus left. It needs to be stabilized. So that is going to interact with the COO minus of the aspartic acid, which is the amino acid. ये आपका DNA polymerase है. DNA polymerase is made up of proteins. they have got the amino acid. So that amino acid is your aspartic acid. So COO minus, COO minus. So they are helping your metal ion to be stabilized so that they can interact with the beta gamma phosphate to stabilize them. Okay? And in between, wherever the electrostatic interaction is not possible, in order to stabilize everything, hydrogen bonds will be present. आगे जाके जब आप देखोगे active site catalytic site में, if you don't have metal ions for the stabilization, then you are going to have the hydrogen bonds. Okay, so overall these weak interactions are making the successful catalytic site for the enzymes to work. अभी थोड़ा सा clear हुआ? See, to be frank, I'll tell you one thing. During my CSR exam, it was the written exam. And this question has come. Plus, there was another question like how, that is like in the translation. Okay, वो translation related question था कि कैसे EPA रहता है, right? If you remember in the ribosome exit and the peptidyle and the new incoming amino site is there. So how from A to P it is being shifted and how from P to E it is going in the ribosomes. वो we needed to explain with the help of the this much space was given to write. What I did, I just draw the diagram. मेरे वो easy पड़ा. And it was given in the textbook very beautifully in the Watson itself. So what you can do is, for your better understanding, just try to draw this figure. Okay. The problem is coming because we forget our basics, what we have studied in 8th, 9th standard. Just draw this once and you can see, you can make the interactions. Plus minus के बीच में जो रहेगा वो आपका electrostatic interactions रहेगा. So just study all these. Okay, just try to read. So you can see how they are helping in stabilizing the phosphate. Okay, later on what will happen? The bond is broken over here. We have got the PPI, and in the previous lecture we discussed pyrophosphatase will come, which will convert into 2PI. So in the nucleus we have got the phosphate which is present in the pool, readily available for the catalysis. Yes. अभी थोड़ा सा clear हुआ? एक बार diagram बना के देखना. It becomes easy to understand. Yes. Anything else? Now look at the figure over here. This is how your crystal structure looks like. Are you able to correlate the same thing जो अभी हमने discuss किया? Can you see the metal ions over here? The green color metal ions. दिख रहा है क्या आप लोग को यहाँ पे? One and
Yes, ma'am.
yeah. Now this colored is your DNA polymerase. Okay. This gray is your DNA. Can you see the helical? The dark gray is the template and the light gray is the primer. Okay. Red आपका हो गया O, OH. Yellow हो गया आपका DNTP. कहीं पे भी, whenever in future also, whenever we are going to study about the crystal structure, always remember wherever oxygen is present, it will be given in red only. ठीक है, बाकी सारी चीजें चेंज होती रहेंगी, बट जहां पे भी ऑक्सीजन रहेगा वो आपका रेड कलर में हम शो करते हैं. थोड़ा सा रूल्स होते हैं क्रिस्टलोग्राफी में, like how to show the structure and all. Okay. Now, just imagine how exactly this will be happening in the nucleus. So you have got the, now look at the structure of the DNA polymerase. Do you think that it looks like a hand? Yes, मुझे तो नहीं लगता, but ठीक है, I have accepted the concept that it is like a right hand, but to me it does not look like a right hand. Anyways, okay guys, और कोई doubt है आज के lecture में? Because I'll be stopping here now.